Thank you very much. I want to say, first of all, that I'm truly honored to be able to speak in this fantastic and amazing set of conferences that are occurring this week. It's just great, and it's very exciting to hear about all the wonderful work. In particular, though, I also want to thank Bengt for his work and his intellectual leadership in organizing Nobel conferences and symposia over so many years. I was privileged to be able to visit these in 1999, 2001, 2008, and now another wonderful one. So I'm going to talk about single molecules and blinking in photocontrol and how that connects to this super resolution microscopy idea. And in order to do this, I first want to take you back to 1985, the middle 1980s, when so many new single objects were being observed and studied. For example, single ions and traps and that sort of thing. And the question was at that time, can you observe single molecules? But there was a problem. Erwin Schrödinger, one of the great founders of quantum mechanics, said we never experiment with just one electron or atom or small molecule. And that he wasn't the only one who felt that way. Basically, it was thought to be impossible. So the first part of my story requires me to explain to you how we got to the point to believe that it was possible. And to do that, we have to go back and look at spectroscopy of molecules in solids at low temperatures. So let's take, for example, suppose you cool down something like this pterylene molecule in a transparent host of paraterphenyl and go to 2 degrees Kelvin, everything narrows dramatically. All phonons are removed, vibrational modes are not excited, etc., and you get a very narrow electronic origin because this is a relatively flat, rigid molecule, and it's the pi electrons that are giving this transition. The absorption, the first electronic transition, gets so narrow that you even see different sites in the crystal. These are different inequivalent locations in the crystal for that first electronic transition that's shown right there. So let's look at even higher resolution with a better spectrometer and so on in the visible. Switch to pentacene. It's another molecule of similar electronic behavior. And here's 2 of the sites for pentacene and paraterphenyl, where now they're, quote, resolved, unquote, but unfortunately they're still a little bit too wide. In particular, these are about a wave number in width, but at this low temperature, they should have been far, far narrower because the excited state lifetime is about a nanosecond. These should have been lifetime limited lines. So what's going on? Well, it turns out this is a phenomenon called inhomogeneous broadening. It's very strong at low temperatures for these situations, and that absorption profile is something like this, many narrow lines that are spread over a range of frequencies because the different local environments cause the molecules to shift to different frequencies. In order to observe the homogeneous width inside this line, you have to use some special techniques. For example, you could use photon echoes, but people in those days, in the mid-80s and late 70s, started to use spectral hole burning. And I'm describing this because it's kind of important to see the background of all the early work. In spectral hole burning, you take this inhomogeneously broadened line and you bring in a single frequency narrowband laser and excite only one sort of homogeneous group. Now, if you have some transformation that occurs, some sort of photochemistry or photophysical change that causes those molecules that were pumped to move to another wavelength, what's left behind is a dip called a spectral hole. And the idea back at IBM, where I was working at that time, was to use these spectral holes for optical storage, to tune the laser, and at different frequencies of the pumping laser, you either write a hole or you don't write a hole, and in so doing, you can record ones and zeros. This was one of those great industrial research labs like Bell Laboratories and so on. At IBM, you could explore such an idea for an application, but also do the science behind it and ask deep questions. For example, what are the ultimate limits to this frequency domain idea? And that's sort of intrinsically linked into all this science associated with inhomogeneous broadening and the molecules producing the absorption and so on. Well, in particular, I wanted to ask that at those days, in the 80s, this fundamental question is there some spectral roughness that results from statistical number fluctuations coming from the discreteness of individual molecules? So the question is this line smooth? In fact, I showed you some real data there. Okay, are these smooth profiles, or do they have some intrinsic roughness on them? And so to detect that, we used a technique called FM spectroscopy and took this pentacene and paraterphenyl system. And and I cooled it to low temperatures. And now imagine that inhomogeneously broadened line is really broad. We've spread it out with very high resolution, so it's essentially flat on the scale of the stage. And what do you see? You see this amazing spectral structure. Here it is. Here it is where we're showing a broader range. But if you zoom in, you see this amazing behavior structure. We call it statistical fine structure. You scan the same region again, you get exactly the same structure. This is not noise. That's why I'm calling it spectral roughness. Well, it's coming from the fact that at each new homogeneous window of frequency space, while there may be an average number of molecules that are in molecules at any given homogeneous width, in fact, that number should fluctuate from bin to bin according to number fluctuations. And therefore, from bin to bin, you should see changes that have a RMS size that's the square root of the number of molecules in resonance. Really interesting spectral effect. That is, this signal's amplitude scales as the square root of the number of molecules in resonance. Now, it sounds so high resolution, so esoteric and all that, but in fact, it's beautiful because it's coming from the single molecules underneath this absorption line. And so it arises directly from that discreteness, and observing this for the first time gave us the hope that we could detect single molecules. Why? Well, if the average absorption here is coming from 1,000 molecules, then the amplitude of this is about a square root of 1,000, or about 32, and so we only have to work 32 times harder to see a single molecule. Not 1,000 times harder, you see. It's much better. Scaling. So that's why we stayed with this method, FM spectroscopy, and the next postdoc, Lothar Kador, and I used FM to push to the individual molecule limit in 1989. And so here's the scheme. Here's an absorption of a single molecule. Here's two copies that are produced by FM. Here is the derivative. We were using a modulation method for technical reasons, so a single molecule should look like this W, and you can see it right there. So this method, FM, which was invented by Gary Bjorklund, is insensitive to scattering. Scattering doesn't matter. We had a cruddy sample that was sort of cleaved and had rough surfaces. You still see this because you're detecting it on the transmitted light coming through the sample. Limited by shot noise, and in principle, since it's shot noise limited detection, you could detect very, very small features. But we couldn't make the intensity large enough to achieve that kind of sensitivity level, because we would broaden the molecules if we turned up the intensity. So anyway, this is very much like FM radio at 506 terahertz, and it showed us that penacene and paraterphenyl was an important model system for getting started in all this. So a year later, Michel Horry made a crucially important advance by also detecting the absorption, but doing it by detecting the emitted fluorescence. So here's his data. Here's the inhomogeneously broadened origins that I talked about. And here down at the bottom are these little dots are the individual molecules detected by measuring the fluorescence coming from the molecule. This method, of course, is sensitive to scattering from the sample. So you have to make extremely clear samples that don't have background or excessive Rayleigh-Raman scattering and so on. But it does give you better signal to noise for equal bandwidth. So the whole field moved to this method afterward. It's basically an application of LIF, laser-induced fluorescence, in gases that Dick Zare pioneered in 1968. Okay, so now with that method, let me show you some of the things that happened in the early 90s. And here's Pat Ambrose's data of penacene and paraterphenyl, one of those inhomogeneously broadened lines. You can see the fine structure now very well. Zoom in, just look at one of them. The width is only 7.6 megahertz at a center frequency of 506 terahertz. So this is incredibly high Q, okay, nearly 10 to the 8th. And very narrow spectral features are wonderful for the spectroscopist, of course, because they're very sensitive to strain fields or other effects, Stark effects, and much was done in the science of single molecules at low temperatures. We also started doing imaging very early on. This is tuning the laser on this axis to select molecules in frequency space, but moving the laser spot on this axis. So it's in one dimension, one spatial dimension. And the single molecules are these little mountains, which we realized even back then that we're using the single molecule to measure the shape of the laser spot. We've got this 1 nanometer, roughly 1 nanometer sized object, and we're moving the laser beam over it and measuring that shape. Very soon after these measurements, this extended to two-dimensional microscopy in Norris Field's lab, and so on. Well, jumping into this new regime of being able to detect this ultimate object, a single molecule, opened up the possibility for observing new, unexpected effects. So let me show you a couple of those. First of all, here we're going to be scanning over frequency space again and again and again and again, looking at penacene and paraterphenyl. And that's one molecule that's doing some sort of really crazy dance. It's not sitting at one frequency, but remember, we're at 2 degrees Kelvin and we're in a crystal. What's going on? This was really exciting to see at that time. If you imagine I put the laser at one frequency, then I would have signal, but as this molecule jumps into and out of resonance, it would be sort of blinking on and off. And this motion in frequency space turns out to be due to nearby host dynamics. Even in the crystal, there are some low-frequency excitations that can be pumped at 2 degrees Kelvin. So that's one of the surprises. In effect, molecules could jump around. Another thing that happened here, Thomas Bachet switching to perilene and polyethylene. Let's scan a single molecule. It looks quite stable, no surprises, kind of boring. But if you bring the laser into resonance with the molecule and just sit on the top of it, this emission signal will go down. And if you scan, you see the molecule's gone. It moved somewhere else. It moved way out off the screen in frequency space. And then you wait a little bit longer and scan some more, and you see it's come back at exactly the same place. We scanned it multiple times, come into resonance, drive it away. So we could switch the molecule back and forth from one frequency to another and measure the kinetics of this process, which was Poisson distributed and so on. But this was the beginnings of being able to switch single molecules. So those are some interesting surprises that occurred in those low temperature days. Well, now we're going to switch to room temperature, of course, and I just want to take a short break just to point out why we find this area of science exciting. When you study single molecules in complex systems, let's say in crystals or in cells or in polymers or so forth, you have this property of being able to remove ensemble averaging for sure. But then you can explore, is there any heterogeneity? That is, are the different molecules the same or are they different? Are they marching to different drummers? You can follow state changes, test theoretical understanding, and so on. And you can image using the fact that these are nanoscale light sources. Using FRET, you can measure relative distances on a very small scale. And of course, super resolution is one of the reasons, another reason that we'll talk about in a second. And there are commercial applications now too. So at room temperature, all of the measurements that are done use this kind of setup, and I'm going to describe, just to make sure everybody's on the same page, how we detect the molecules at room temperature now. But let me just point out, in the 90s, there were many people who made important steps forward in terms of observing molecules in solution, the bursts of fluorescence as they move through the focal spot, near-field imaging, confocal imaging, wide-field imaging with total internal reflection, and so on. In other words, basically any method that you could think of worked for detecting these single molecules. So how does it work? Well, we're pumping from the ground state to an electronic excited state, and after vibrational relaxation, collecting this red-shifted fluorescence. We tend to avoid dark states, but you can use dark states, as you'll see in a moment. Focusing down to the smallest spot possible due to the Abbe diffraction limit of about lambda over 2 Na, or 250 or so nanometers. In order to achieve the single molecule limit at room temperature, you have to make sure the molecules are far enough apart. You have to dilute, make sure that they're far enough apart beyond the size of this spot. Remember, at low temperature, we could just tune the laser color and select the different molecules. Here, you have to just simply dilute by brute force. The experiments involve attaching labels to the molecules of interest, and this can be done in cells or in many other environments, and here's some examples of these floors, as you've seen. Well, if you just do this, that is, if you dilute and watch individual molecules, in the late 90s and early 2000s, many things were done, many interesting experiments were performed, and I just want to quickly, very quickly, sort of just give you a quick flavor of what can happen in one wonderful experiment that we did with Hardin McConnell, bless his soul. Unfortunately, we lost him last year. We were looking at diffusion of transmembrane proteins. This is MHC2 proteins on the surface of a cell, and this beautiful diffusive process, which is dancing of those transmembrane proteins on the surface of the cell, gives you access to information about the membrane and the cholesterol concentrations and so on. Another class of behaviors, rather than diffusive behavior, just look at what this molecule is doing. Just watch where this arrow is. You see that it moves in a line across the cell and then goes around the backside of the cell, also in focus. This is directed transport. Something's moving that molecule for a reason. It's no longer random diffusion, and that's because this molecule in this bacterium has to do with the cell wall machinery. And to give you a material science example, let's just go back to terylene and paraterphenyl at room temperature. Okay, so now we're room temperature, and here's the image of the emission from all these molecules. If you look closely, you see that some of them have little rings. That ring is coming from molecules whose dipole moment is pointing out of the page, pointing out of the screen towards you. In other words, if the dipole is along Z, then the emission is this big donut, and it looks like a little ring. So the shape of the image tells you the orientation of the molecule. But the movie tells you even more. If you watch the movie, you see that not only do you have those little rings that aren't moving at all because they're in fixed parts of the crystal, there's other single molecules that are zooming around, even at room temperature. And there's no membrane or anything here. It's a crystal, but in fact, it's not a perfect crystal. It has cracks, and these are the molecules that are moving in the cracks. It's very biased diffusion. They move mostly up and down or along another crack direction. And so the motion of the molecules tells you about the properties of the sample, in this case, where the defects are, where the molecules can move around. Another thing that happened in those days of watching single molecules at low concentration was our discovery that you could blink and switch single copies of fluorescent proteins. This was those beautiful days of early work when Roger Chen was using green fluorescent proteins for labeling in cells. And we asked him to give us some of this particular yellow fluorescent protein mutant so that Rob Dixon could try to see if you can observe these at room temperature. And you can. Here's an image of the YFP, yellow fluorescent protein at room temperature. And once again, the surprises started appearing. These molecules blinked as well. Even at room temperature, you see it on, and then off, and then back on again, and then off again, and so on. Meaning that we're cycling the molecule and detecting photons, and at some point it enters into a dark state, goes over a barrier. This is probably isomerization of the GFP chromophore, from which it can return thermally and start emitting again. We also saw that if you wait a little bit longer, you see molecules apparently photobleach, that is turn off, we thought for a long time, but if you use a little bit of blue light, you can turn those back on as well. In other words, here's a molecule that's been used up, and after a little blue light, it's restored, and you can use it again for a while until finally it turns off and on and so forth. So this control again appeared at room temperature here, even with these interesting fluorescent proteins. And after this, a variety of experts started refining and generating mutants that have enhanced properties. I show some of them here, which are important. So let's go on now to super resolution microscopy. Because now that I've sort of set the stage and talked about these fundamentals, I want to connect how all that early work merges into the things that we're doing today. You heard about stimulated emission depletion microscopy from Stefan, his beautiful idea. Another method for super resolution that does not require single molecules is something called SIM, structured illumination microscopy of Mats Gustafsson, and a number of people are using that method, especially Eric Betzig because he likes it. But I'm more interested today in talking about the approaches that do require single molecule imaging, and this is how I connect more directly to the third laureate, Eric. So the question is, how can you use these single molecule labels to surpass Abbe's diffraction limit? And the answer is single molecule imaging. That's a key requirement. Plus you have to add on two key ideas. And just to make sure there's no confusion, our contribution is on the foundations of these ideas. So let's first talk about the two key ideas. The first one is what we call super localization. Now, how does that work? Well, imagine this beautiful little cinder cone. This is in Crater Lake, Oregon, and if you haven't been there, I urge you to go. It's incredibly beautiful, a lake with this little cinder cone in the middle of it. And here's my scale bar, 120 times 10 to the 9th nanometers, okay? Got to have your scale bar, though, right? So you know that if you take your cell phone and you walk up to the peak of this mountain, you can record the coordinates of the position of the mountain. That's basically what we're doing with single molecules. You find the position by fitting the shape of the single molecule image. So let me explain. Here's one of these images of single molecules now, X and Y, and we're looking at the light coming from the molecules. They look like little disks, but they're not disks. If you spread out that light out over many pixels of the detector, you see that there's different numbers of photons on the different pixels. In one dimension, this has a shape. That's coming from the fundamental response of the microscope, the point spread function of the microscope. And in particular, we know now that the different pixels are sampling the shape of that point spread function. Is an Airy function for conventional microscopes, and that can be approximated very well with a Gaussian shape. So the idea is to use a fitting function, a Gaussian fitting function, which has a width that is still diffraction limited, but the estimates of this other parameter of the function, the center position of the function, will have a much, much smaller error distribution than the diffraction limited width. That is, it'll have a localization precision, we call it, sigma, that scales according to the Abbe limit divided by the square root of the number of photons that are detected. In other words, each photon landing on the camera is like a position measurement, and you've done n measurements. That's why your precision can improve this way. So if you have 10 to the 2 photons, then you get a factor of 10 below the diffraction limit, 10 to the 4th photons, a factor of 100, and so on. Okay, great. So this is localization. But you can see it only works for a molecule that's isolated. When molecules are close together, we have the resolution problem. You have to be able to distinguish molecules that are close to one another in order to have true resolution. And so in 1995, Eric wrote a really important paper that said, well, I think I can solve this problem, the theory paper, if I have another variable that lets me separate those different molecules along another dimension. And if I can do that, then I can make two molecules that are overlapping be distinguishable. And so that was a key idea, but it wasn't experimentally demonstrated until some scientists used the low temperature tunability that I talked about earlier to achieve super resolution using that idea. But that was only a few molecules, and so the truly general idea appeared a little bit later. So that's key idea number 2. And I'm going to describe it by calling it active control of emitter concentration and sequential imaging. So let me explain. If you have no active control, okay, then and you start out with a structure like this, you first then decorate it with these fluorescent labels, as we talked about earlier. And now, if you turn on the laser, then if you allow them to all emit at the same time, then you get this blurry image. You get all of the point spread functions are overlapping, and that's been the problem of resolution in the visible up until all of this work on super resolution. So you can see the solution now. Don't let them all emit at the same time. It's pretty obvious when you explain it this way. And you can use these tricks of having molecules that are in dark states and in bright states and so on that allow you then to, as an experimenter, to actively make a choice to make sure that the concentration is not large at any time when you're doing the imaging. In other words, suppose these molecules are photoactivatable. Suppose they don't become fluorophores unless you give them a little tickle with some control light. You can do that with arbitrarily low intensity, and so only a few will turn on. Now you can superlocalize these and then photobleach them. And do it again. And some other molecules will turn on. And if you do this multiple times, then by sequentially sampling different positions under the structure, you can take all the positions that you determined and in a pointillist way reconstruct the underlying structure. So that's how it works, to get information beyond the Abbe limit, using those beautiful little molecules as tiny light sources that are decorating the structure. So this idea I first heard about from Eric Betzig in April 2006 when he was giving a lecture at the NIH in April 2006, and he called it PALM, Photoactivated Localization Microscopy. Very quickly after that, a storm appeared from Zhuang, F-Palm, and then things called PAINT, I'll talk about later, D-Storm, GSDIM, Blink, whatever. So basically, all kinds of people realized, hey, I can come up with another way to achieve this active control. So many of these acronyms refer to different ways of achieving active control. Well, we did something then called YFP reactivation, and we didn't use any acronym, of course. So that was forgotten, right? There was no acronym. So to resolve that problem, then I'll give you another acronym just for fun, a mechanism-independent one, single molecule active control microscopy, or SMACM. Okay. All right. So let me illustrate then a few ways that this appears in different experiments. Let's use the blinking method, okay? Let's use molecules that blink a lot, and if we set up the intensity correctly, then most of them will be off and only a few on. So this is one of the things you can do if you use YFP, again, yellow fluorescent protein, fused to a bacterial protein, and we're doing this in Caulobacter crescentis, a collaboration with Lucy Shapiro. Here's the different cells, okay, in white light, and here's the fluorescence image, and here's the fluorescence movie. You see that in each frame, these YFP fusions are emitting and then turning off, emitting then turning off, and we're working in that regime where there's only a few on at a time. So you can use superlocalization to find all their positions and then reconstruct the underlying structure. That idea is what we've been using for a while in bacteria, and other people are using it as well. Here's the epifluorescence diffraction limited images for 3 different proteins in the bacteria, and here are the super resolution images of those same proteins. So it's this difference between these fuzzy images and these higher resolution images, which have a resolution of or precision near 40 nanometers or better, show incredible detail that wasn't visible before. That's why people are so excited about the super resolution microscopy, by whatever method that you like. To illustrate a different method of active control, I want to switch to this paint idea. There was an idea that was put forth by Robin Hochstrasser also in 2006, and I'm mentioning him in our memory of him, another great physical chemist that we've lost. But the paint idea involves this. You put the labels in solution outside of the structure that you want to image, and they zoom around by diffusion very, very fast so you can't see them. But when they bind to the structure of interest, you can see the light from those fluorophores as a bright spot of light. And so you can make sure that you have low enough concentration just by whatever concentration you use and what power you use. And so we've used that paint idea in a collaboration with one of my synthetic friends, Justin Dubois. Justin can make this molecule saxitoxin, okay, a synthetic, difficult synthesis, but certainly doable. And in so doing, he can add a fluorophore to it. So this fluorescently labeled ligand is the molecule that zooms around in solution outside of, in this case, a cell. We're going to use saxitoxin, since it's a neurotoxin, to light up voltage-gated sodium channels in a differentiated PC12 cell, which is a neuronal model cell. And this is after causing the cell to differentiate. The body is off scale, off the screen. This is one of the axonal-like projections. And each of these spots is a position that we found by this microscopy that I've been talking about, where saxitoxin bound to voltage-gated sodium channels. We can even do a little bit of time-dependent imaging here, because one issue about these single molecule localization microscopies is that you have to time sequentially, like time domain multiplexing, image the fluorophores at different times. But people are pushing the speed to higher and higher speed, and even here, we took every 500 milliseconds of this image, every 500 milliseconds of the data, and display it, and then do each new 500 milliseconds in a sliding window. So if you do that, then you can produce this time-dependent set of images. And so remember, each spot is lighting up a voltage-gated sodium channel, and you can easily see neuritic extensions that are extending out from this axonal-like structure at many locations along the cell. So that's just an example of paint, and paint actually is being used for looking at DNA and origami and many other things now. A third example, looking at some important medical problems, in this case Huntington aggregates. It turns out the Huntington protein, when it is mutated by expansion of the polyglutamine region, so that you have a large number of Qs, large number of glutamines in a row, then aggregates form in Huntington's disease, a serious neurodegenerative disease. So we started looking and have been looking at individual
fibrils by looking at a cell system where the Huntington has been fused to YFP and then using the YFP blinking to image the shapes of these fibrils. One thing to remember in a lot of these neurodegenerative studies, once you have aggregates, mostly what many people see is a huge inclusion body. That is a kind of a large object, diffraction limited. Here's one of those inclusion bodies. But we're more interested in all this other little stuff that you can't normally see unless you use high sensitivity and super resolution methods. Those are small aggregates that here in super resolution, reverse contrast, you can see they're very tiny. And even out in an axonal projection, you can see these structures. Here's a diffraction limited image and a super resolution image of Huntington fibrils. This done by Stefan Zoll, a postdoc in my lab. So I think there's a lot of future for that as well. Now, in the last few moments that I have, I would like to talk about some of the other areas where this field needs to continue to expand, and give you some examples of that. First of all, we need new fluorophores. We need better fluorophores. We need more switchable fluorophores. We would like to have more stability and more photons from the molecules. A very simple example with Bob Twigg, a synthetic chemist, we took a look at these rhodamine spirolactam molecules because of the interruption in the conjugation. This is a closed isomer and it's not fluorescent. But a little bit of blue light will open the lactam ring and produce a rhodamine dye, a bright fluorescent molecule. So you can see this is an active control scheme that you could use. All you have to do is to make sure that you only dimly activate the fluorophores. And by taking these molecules and binding them to the surface of bacteria, binding them through an NHS functionality to the surface lysines, we light up the surfaces of these bacteria and can do super resolution images to see all of these stalks, which are far beyond the diffraction limit. So that's just an illustration, but what's really needed is more and better fluorophores of this type. Another area of great interest these days is three-dimensional imaging, extending all these images that I've talked to you about from 2D into 3D. And there's a number of ways to do that, but we've been doing it by this scheme. We convert the microscope from having the standard response to the double helix response. Using some optical wizardry, essentially, we make the image of a single molecule a pair of spots. And this pair of spots, depending upon the Z position of the molecule, will have a different angle. They basically revolve around one another as you move a molecule up and down in Z. That's why we call it double helix, because the point spread function of this microscope looks like a double helix along the Z-axis. Using that scheme and using this molecule, then you can get these three-dimensional images of these Colobacter crescentus cells, where this molecule is certainly densely labeling the surface and giving us plenty of photons to have a high-resolution image. Now, at the last thing that I want to talk about today is I just want to remind everyone that single molecules themselves are also interesting. Of course, super resolution is really exciting, and there's all kinds of interesting problems to work on to make super resolution better and better. But just remember that if you go back to individuals, you still can do neat things with these ultimate small objects. I want to briefly say how we're doing that and what we're doing for single molecules that are in solution, that are in a native solution environment. And we've been using something called an Abel trap, an anti-Brownian electrokinetic trap, which is a way of confining single molecules using closed-loop electrokinetic feedback. It's basically a microfluidic-like cell where we are trying to trap the molecules in the center of the cell, and we use voltages that we apply to the solution to impress forces, electrokinetic forces, on the single molecule. And essentially, whenever it moves off the center, you put on a force that pushes it back, and if you do this at high speed in a closed loop, you can make the molecule just jiggle around the center. Well, this is a great way to study molecules that you don't want to attach to the surface, you don't want to tie down, you don't want to perturb, and we've used it to look at all kinds of photosynthetic proteins and single enzymes and other structures. It's basically inferring information from the fluorescence of the molecule. But in the most recent work, a student, Chen Wang, has gone even further. So in one of these trapping experiments, remember that we measure the intensity of the object, and you can see it looks relatively constant here, fluctuating a little bit. But we also know information about the position of the object, the position of the molecule that we've been using for the feedback. And we also know the voltages that were applied to try to keep it near the center. Well, if you think about this, there's an underlying Langevin equation that's controlling the motion of the molecule. And so we would like to infer D and mu. So by a really powerful Bayesian learning algorithm, Trinh figured out a way to extract maximum likelihood estimates of D and mu. D and mu, those are physical properties of the molecule. D depends on 1 over the hydrodynamic radius, and mu depends upon the charge on the molecule. So for every 50 milliseconds, we can estimate on the fly the diffusion coefficient and the mobility of the single molecule that's trapped. So if you take that information, then you have access to other biological and biophysical parameters. For example, you can look at dimerization and tetramerization and oligomerization in general. This is data now in a mu-D plot. Each black dot there is a single measurement of D and mu by the method I just showed you. And tetramers scatter in this range, but monomers scatter in this range. We can measure on the fly whether we have what fraction there is of monomers and trimers in this particular case, because it's a photosynthetic protein. Another way you can use this is to measure binding interactions. Here, if you take a single-stranded piece of DNA and fluorescently label it and trap that tiny object, then add unlabeled complement to the solution, there will be formation of hybrid and melting and so on. But when this happens, D and mu move oppositely from one another. When you form the complement, D goes down, but you add more charge, so mu goes up. So D and mu, in real time in this experiment, while the intensity is constant, D and mu are toggling back and forth in this anti-correlated fashion, corresponding to the single binding and unbinding of this single strand of DNA. So this just shows you that this can be used to look at protein-protein interactions and all kinds of other effects. Okay, so to sort of end up, I just want to say that single molecule spectroscopy and imaging have been affecting many fields of science. It's wonderfully interdisciplinary and great fun. And working at this limit has attracted the attention of many, many talented people all over the world. And their contributions, of course, are the key seminal components that has pushed this field forward so much. I haven't had time to talk about all the other neat things that have happened, Fred, enzymes, actin bands, and RNA, and all kinds of other processes, but I think you see the idea that it's an exciting time for single molecules. I want to extend thanks to the current group of students and postdocs. We call ourselves the guacamole team, by the way, because as you know, one molecule is one guacamole. It's 1 over avocado's number of moles, right? So it's such a bad joke, but you know it's so good that you just can't avoid it, right? Thank you for laughing. We have a no ensemble averaging logo as well, and so I want to also extend thanks to past, present students and postdocs and collaborators and the committee, mentors, institution, and family. This took 5 slides, you know, during the Nobel lecture. I don't have time for all of those, but I want to thank the agencies, of course, because that's our lifeblood. And finally, I want to thank you for your attention and say once again, happy birthday, Bengt.
Thank you for a fantastic presentation, Professor Moerner, and teaching us that if you don't have an acronym, you don't exist. And also the evolution of hairstyle during your career. Was interesting to see.
Yeah.
I think before coffee we can allow one very quick question if there is one. So we have one up there.
Yeah, you told us that in order to get a 2D image you have to have sequential images, and then if you want to go to 3D, this is even one step further. So I wonder if you want to look at live systems, what the time resolution would be that you can resolve.
So it's a great question. So in order to do these experiments, in order to collect information about the molecule's positions, we have to detect photons. So if you want to go faster, then you want to have the molecule emit photons faster. So you can control that by the intensity. And turning up the intensity, you get more photons per second. So you're correct that trying to go to 3D means you have to get more positions and more localizations. But remember, we don't have to do any sort of sectioning. This is not our 3D methods are not confocal-like in terms of confocal sectioning, where you have to get different layers at different times. We get all the molecules all at the same time using these double helix and other schemes that let us just measure X, Y, and Z simultaneously. So what's been going on to address your problem is to various people, like Jörg Buersdorf has pushed it so that you can go faster and let's say take some images in the time scale on, let's say, half a second or 100 milliseconds or something like that, 10 milliseconds. It's been pushed down to sort of that time scale. And so there's been, progress and a lot of progress in that area.
Thank you.
Okay, let's thank Professor Moerner again.